A baby is born. He looks healthy. But inside his body, a silent process has already begun. A toxic chemical is building in his blood. It's heading for his brain. And his liver, the organ designed to stop it, is missing a single instruction. One letter wrong in 3 billion. That's all it takes. But in 2025, a team of scientists did something that has never been done in the history of medicine. They went inside this baby's body, found that one wrong letter, and changed it. This is the story of baby KJ, the first child to receive a personalized CRISPR therapy inside his own body, and what it may mean for the future of rare genetic diseases, precision medicine, and brain health forever. Right now, your liver is doing something remarkable. Every time you eat protein, a steak, a handful of nuts, a glass of milk, your body breaks it down, and that process creates a waste product. Ammonia. You've probably smelled it in cleaning products. It's harsh, caustic, toxic, and your body makes it constantly. So your liver runs a detox process called the urea cycle. It converts ammonia into a harmless compound called urea, which you flush out in urine. You never think about it. It just works. But what if it didn't? In a rare genetic disorder called CPS1 deficiency, the very first step of this cycle fails. The liver is missing an enzyme, carbamoyl phosphate synthetase 1, that kicks off the entire detox process. Without it, ammonia doesn't get neutralized. It builds. It enters the bloodstream. It crosses into the brain. And when ammonia reaches the brain, it causes swelling, seizures, coma, permanent damage. In severe cases, this can happen within the first 48 hours of life. CPS1 deficiency affects roughly 1 in a million births. For most families who face this diagnosis, the path forward was brutal. Strict low-protein diets, powerful medications, and hope that their child could survive long enough to receive a liver transplant. But newborns are often too small for transplant surgery, so parents wait and worry. Until very recently, that was the ceiling of what medicine could offer. But what happened next surprised even the scientists. Because fixing DNA inside a human body had never been done like this before, and the tool they used wasn't the CRISPR most people think of. In 2024, a baby boy named KJ was born in the United States. Within days, doctors confirmed he had severe CPS1 deficiency, one of the most dangerous urea cycle disorders known. For KJ's family, time wasn't just ticking. It was running out. Every hour carried the risk of rising ammonia levels. Every spike increased the danger of permanent brain injury. But a group of researchers from Children's Hospital of Philadelphia, Penn Medicine, and gene editing teams around the world looked at this baby and asked a different question. Not how do we manage this disease, but what if we could just fix it? They already knew the exact mutation causing KJ's disease. One incorrect DNA letter in his CPS1 gene. One typo out of 3 billion genetic letters. So they set out to build a therapy specifically for him. Not a mass-market drug. Not a treatment designed for thousands of patients. A personalized CRISPR gene editing therapy for one child's genome, and they built it in 6 months. To put that in perspective, the average drug takes 10 to 15 years to develop. KJ didn't have 15 years. He barely had weeks. Scissors versus pencil. How this new CRISPR works. Most people have heard CRISPR described as molecular scissors. The idea is simple. CRISPR finds a specific place in DNA and cuts the strand, allowing cells to repair the break. But what KJ received was something even more precise. It's called base editing. And the difference matters. Traditional CRISPR is like scissors. You cut the DNA and rely on the cell to repair itself. Base editing works more like a molecular pencil. Instead of cutting the strand, it zooms in on a single genetic letter, erases the incorrect one, and writes in the correct one. No large cuts, no broken strands, just a precise correction of a single typo in 3 billion characters of code. But there's still the delivery problem. How do you get this molecular pencil inside the right cells inside a living baby without surgery? The answer: lipid nanoparticles. Tiny fat-based bubbles, barely visible under a microscope, loaded with the CRISPR base editor, the same delivery technology used in mRNA vaccines. Injected into the bloodstream. The liver, because of how it filters blood, naturally absorbs them. Inside those liver cells, the base editor locates the CPS1 gene, finds KJ's specific mutation, and corrects it. For the first time in his life, KJ's liver cells had the correct instructions. Before we get into the results, and they are genuinely worth staying for, take 2 seconds and hit subscribe and turn on notifications. On this channel, we break down breakthroughs in brain health, genetics, and neurodevelopment in a way that's clear, hopeful, and practical for real families. No jargon, no fluff, just the science that actually matters. It takes 2 seconds, and it means you won't miss what's coming next. Okay, back to KJ. KJ received his first infusion in February 2025. Then a second dose, then a third. Each time, doctors watched carefully. They measured ammonia levels, metabolic markers, how much protein he could safely tolerate, any signs of side effects. And the results came back. His ammonia levels stabilized. He began tolerating more protein in his diet, something that had previously been dangerous for him. And doctors did not observe severe side effects from the treatment. A child who once lived on the edge of constant metabolic crises began showing real, measurable improvement. Researchers described it as the first personalized in-body CRISPR gene editing therapy ever administered to a human being. Not in a petri dish, not in an animal model, in a baby boy named KJ. This isn't about one baby. It would be easy to hear this story and see it as a one-time miracle. An extraordinary case, exceptional resources, a lucky child. But scientists see something else entirely. There are over 7,000 known rare genetic diseases. Most are caused by mutations in a single gene, just like CPS1 deficiency. And most still have no cure. Not because the science is impossible, but because of a brutal equation that has governed drug development for decades. Developing a new treatment requires clinical trials. Clinical trials require thousands of patients, and diseases that affect 1 in a million people can never generate thousands of patients. So they get ignored. Too rare, too complex, too expensive. That's been the cruel math of rare disease medicine for generations, and it has left millions of families with the same devastating sentence: there's nothing we can do except manage it. KJ's case challenges that entire model. If scientists can design a CRISPR therapy for one specific mutation in one specific child in 6 months, then medicine may be entering a completely new era. a future where treatments aren't designed for populations. They're designed for individuals. Scientists call this N of 1 medicine. Treatments built for a single genome. For decades, that idea sounded like science fiction. KJ made it real. Now, and this matters, science doesn't end with one promising result. There are still hard questions ahead. Will the correction last? Liver cells divide as KJ grows. Researchers need to understand whether edited cells persist or get gradually replaced by unedited ones over time. Could there be off-target edits? Base editing is precise, but no system is perfect. Long-term monitoring will need to confirm that other parts of the genome weren't accidentally altered. And then there's the question that goes beyond biology entirely. Who gets access? Right now, designing a custom therapy for one patient requires a level of scientific coordination that very few institutions in the world can pull off. That has to change for this to matter beyond KJ. But here's what this case proved, and why it matters so much. The biology works, the delivery works, and when the medical system moves with urgency, the timeline of medicine can shrink from decades to months. For families facing diagnoses they were told were untreatable, that shift is not a small thing. Where this is all heading. CRISPR has already begun transforming medicine. Sickle cell disease, certain blood disorders, inherited forms of blindness. But those therapies were developed for conditions affecting hundreds of thousands of people. KJ's case is different. It proves that gene editing can reach the diseases that have always fallen through every crack. Too rare, too complex, too small a market to justify the investment. And if scientists can begin building shared libraries of base editors, delivery systems, and genetic design pipelines, future therapies could be created faster for each new patient. Instead of starting from scratch every time. For parents who have spent years being told that their child's disease is simply something to live with, this represents an entirely different kind of hope. Somewhere right now, a parent is sitting in a hospital room. They've just been handed a diagnosis for their newborn that most doctors have never seen. And until very recently, there was almost nothing medicine could offer them. This story doesn't erase that. But it changes what comes next. Because somewhere in a lab, a scientist now knows that designing a therapy in 6 months is possible. That correcting a single DNA letter inside a living human body is possible. That medicine built for one child, one mutation, one family is possible. CRISPR won't fix everything, but it fixed something. And that changes what we believe can be fixed. If this story gave you hope, or raised questions you're still sitting with, I want to hear from you. Does gene editing make you feel hopeful, cautious, or a little of both? Drop it in the comments. I read every single one. And if you found value in this video, subscribe and share it with someone who needs to hear this story. There's a lot more coming.